Alright, back to our episode 2. So previously we talked about the innate versus the adaptive immune system, how the innate is your first-line responders with like more broad spectrum kind of receptors like your toll-like receptor 4 that can go on to activate complement, which has opsonization, recruitment of neutrophils, and also your membrane attack complex using C9. But also with the adaptive immune system. So the adaptive immune system, on the other hand, has much more specific receptors and has a much more diversity of receptors because they need to fit into that specific antigen. So we're going to be talking about in particular how the B cell has that specificity today. All right. So just an overview of the B cell. Firstly, the antibody is also called an immunoglobulin. Okay. So this is his kind of general life cycle. So you have a B cell, and sorry about my handwriting, but anyway, this is the receptor on the B cell is the antibody. But on the B cell, that receptor is glued to the membrane, but that has a specificness to the antigen. So an antigen comes floating along and just so happens to fit into this antibody, which is a receptor. And this is going to therefore activate your B cell. And with two-factor authentication, i.e. help with your T helper cell, this basically turns your bored B cell, which has been sitting around waiting for ages, into someone who's finally useful at once. So that's a plasma cell. And this guy, he's going to make this antibody loads and loads of times. This is a plasma cell. But unlike the B cell, this antibody is now not membrane bound anymore. It's soluble in the plasma and can circulate around and attack the antigens. There's two differences mainly with the plasma cells. So one is that in order to make all these antibodies, they need a lot more endoplasmic reticulum, and you can kind of see that as more kind of grainy under the microscope. And also, B cells live in the lymph nodes. When they turn into plasma cells, they go to the spleen or the bone marrow, and that's kind of where they live for that memory function. Okay, we said there's two-step verification. So in order for the B cell to turn into the plasma cell, it needs a T helper cell to kind of double check. But also there's kind of a triple checking mechanism, just to kind of triple check. There's two immunoglobulins, so kind of antibodies, on either side that flank the receptor on the B cell, and these are called IgA and IgB. So Ig stands for immunoglobulin, and these kind of sit on the surface. And when your antigen binds to your receptor, these guys also can kind of sense it, and they send a signal as well, letting the cell know that it's okay to kind of turn into a plasma cell. So that's IgA and IgB. Okay. So now we're going to talk about the structure of an antibody, and this gets quite complex. And this, like, I watched loads of videos on this and it confused me back in the day. So hopefully this kind of makes sense. So, and if it doesn't, just watch it a few times because these kind of terms are important, especially when you're understanding later concepts. So we have an antibody, and remember they're that Y shape. They're made of a heavy chain and a light chain. So far not too bad. The heavy chain is kind of the bit on the inside, and the light chain is on the outside. And these chains are held together by disulfide bridges. So here and here, and like here and here, for example. And also, okay, each chain. So we have one heavy chain on this side and one light chain, and on the other side we have another heavy chain which is symmetrical and another symmetrical light chain. And each chain is going to have one variable region and one constant region. So this is the constant region, and we're going to add a variable region. So the heavy chain, this is the variable heavy region, and that's the variable light region. And on the other side we have a variable heavy region and a variable light. So remember, this is symmetrical. So both sides are the same. And so your variable region is your light blue colors and your light green colors. And your variable, same thing on the other side, your light green and your light blue. So this is your variable, this is your constant, this is all your light. So variable light, variable light, variable heavy, constant light, constant heavy, and then constant heavy. Cool. So say we're going to have a different kind of genetic sequence, and that's going to code for a different antibody. That's going to lead to a different variable region. So the constant region stays the same, both the heavy and the light, but the variable region is different. So which colors on this diagram represent the variable region, the bit that has changed to fit a different shape of antigen? It's the red and the pink. Okay, so that's this one is the variable heavy, that's the variable light, and together they kind of are different, they make up a different shape for the antigen to fit into. And say we have another kind of coding kind of sequence, and that leads to a different variable region, which is going to be your orange and your purple in this one. Okay, if we zoom in to the variable region here, which is made of the heavy and the light, we're going to see how it's actually made variable. So the variable region is made of 3 loops of protein, and these are called complementarity determining regions, CDR. You can just call them CDR, CD1, CD2, and CD3. And your light chain is also going to have the same thing, 3 loops of proteins, and these loops of proteins are going to have different genetic sequences, and these basically lead to it being variable. Okay, just to confuse you even more, the bit that the antigen fits into is not just the variable region, it's kind of this whole Y-shaped bendy bit. So the variable region plus the constant heavy and the constant light, but just kind of this bit here. So this bit here. So if we zoom into that bit, that is called the antigen binding site, aka FAB, like the isolate. So we've got the light chain here, the variable light, the variable heavy, it's confusing, the constant heavy and the constant light. So that together makes up your FAB region, and this is the bit where your antigen is going to slot into. And because your antigen binds via van der Waals forces or like hydrogen bonds, whatever. But they're not covalent bonds. But the thing is, so technically it's a reversible reaction. But however, they fit so nicely together. They're so snug and cozy. They don't want to leave together. They don't want to leave each other. So essentially, they're always together. So although technically it is reversible, they're such a tight match that it won't really be reversible in real life. Okay. Oh, by the way, the other thing that I missed is that this straight bit here, so this is called the FAB region, and this straight bit here is called the FC region. Okay, so the problem is that the variable region, remember the variable region is made of your CDR loops. The problem is that every cell in your body has the same genes, right? So therefore, and the same genes should mean therefore it goes to the same proteins, right? So therefore, how do we get this diversity? So the way I think of it is like a pack of cards. Maybe this analogy works for you. So say, in order to make a gene, you need the cards 7, a king, and a 3. And with that combination of cards, you can make a nice antibody. If, however, you have a 7, but a king from a different suit, and still a 3, you're going to have a different sequence. So these are segments of genes, and each segment of genes contains codons. So you're going to have a different sequence of codons. So you're going to have sections of genes like a menu. So for example, this 7 is a menu, and within that you have 4 different things that can be. So likewise, you have a menu of things, and these are called VDJ regions. Okay, so instead of 7, king, and 3, you have a V, a D, and a J, and you're going to choose one of each of those menus, and then combine them together, and you're going to get a different genetic product. So although all of the cells have the same set of cards, they can choose a different combination of cards and make therefore a different combination of genes, a different combination of antibodies. Okay, remember this straight bit is called the FC region, and therefore the other bit, the antigen binding region, is your FAB region, and the variable bit is variable because it's made of loops of protein. Your CDR 1, 2, and 3, and your light chain as well has your CDR 1, 2, and 3. So your CDR, instead of cards, they're coded for by a V, a D, and a J region. So the V is going to code for once you picked a V from your first kind of option, then that is going to code for the first loop and the second loop, and also in some light chain as well. It's going to code for the first loop and the second loop, and then the third loop is coded for by all 3. So all 3 of these together code for the third loop. And that is in the heavy chain. In the light chain doesn't have a D region. So instead of having VDJ, you only have V and J coding for CDR3. So essentially, you can pick different combinations of the V segments, the D segments, and the J segments. And these segments are going to code for different parts. So these are the parts that they code to. And because they code to different parts, essentially because they code for different proteins, these parts are going to have different protein sequences. And therefore, your loops are going to be different. And that is going to make your variable region different, and therefore give it a different shape for antigens to fit into. Okay, this is kind of stupid, so skip this if you're clever, but I think stupid analogies work for me. So the way I think of it is like a menu, right? So you have a V, D, and a J region. So V is for very nice starters, and for the heavy chain, you have over 40 starters to choose from, so that's a lot. The D region is called desserts, so it's like a menu. And there's up to 27, so this carries on, up to 27 combinations. And the J region is for juices. You can have up to 6 juices. So just like with the cards, you can choose kind of one of each set. For this one, there are not 4, but there are 40 to choose from. This one, there are not 4, but there are 27. And this one, there are 6 combinations. And you're going to basically choose one of each and recombine them in a way that makes a unique kind of combination. And that unique combination is a unique protein sequence. And that is going to lead to a unique kind of variable region. Different loops as well. Unique combination in each loop. Okay. So, and remember, okay. And so this choosing of the cards or this choosing of the menu occurs in the bone marrow. So ignore the light chain for a second because it gets a bit complicated. If we're talking about the heavy chain, the V and the Ds and the Js, your menu basically exists on chromosome 14. So within chromosome 14, you have your menu, and the thing that kind of picks things from the menu are called recombinase enzymes. These are coded for by the genes REG1 and REG2, and these enzymes work for looking for that dotted line to cut along, which is called the recombination signal sequence. And when they find that dotted line to cut along, they make a cut in the VTJ regions and then join them together. So just to illustrate this, basically what I was saying is how does your cell know that this is the starter section, this is the dessert section, and this is the juices section? Well, basically, there's sequence of amino acids at the end. Where are we going? The sequence of amino acids at the end. Okay, the sequence of amino acids at the end called RSS sequences. And these mark where the starter section ends, where the desserts section begins, etc. And you're going to have REG1 and REG2 recombinase enzymes that are going to come along and cut one gene from each, one option from each, and then join them together. So your RSS sequences essentially are kind of markers on the menu that divide up the menu. Okay. So again, remember we're focusing on the heavy chain for now. Ignore the light chain for the time being. The heavy chain is the inside one. So ignore what I've written here for a second. So the heavy chain can choose up to 40 V segments, 27 dessert segments, and 6 juices. And these are going to recombine into a particular sequence, and that particular sequence is unique to that kind of cell, and therefore it's going to give it's going to allow the protein to fold in a specific way, and therefore we're going to get a different shape depending on the choice that we've chosen. And if you understand maths, you know that like basically you're getting huge numbers by having like 40 combinations in one and 27 in the other and 6 in the other. You're having a massive number of possibilities. Okay, now I said ignore the light chain. We're going to talk about it as if this wasn't complex enough. In order to generate more diversity, there's actually 2 types of light chain. Now, it's not actually that deep. Essentially, there's 2 types. They're very similar, but, and your diagram is going to look the same, exactly the same. Essentially, though, there's 2 types. There's the kappa and a lambda type. So your body is randomly going to choose, hey, I want a kappa type, or hey, I want a lambda type, and it's going to marry that with a heavy chain. And that is basically just all it is another way to add diversity. So you can have essentially 2 types of the light chain, and you're only going to pick one of them. So if you pick a kappa, that's coded for on chromosome 2. So heavy chain is chromosome 14. Kappa, the kappa version of the light chain, is coded for on chromosome 2, whereas the lambda version of the light chain is coded for on chromosome 22. Now, the only basically different thing between them is that the menu of V's, the menu of starters, is slightly different. So whereas the kappa has 40 things on its menu, the lambda has 30. And remember, the light chains do not have a D region, and the way I think of it is D is for desserts, and because you're going light, you're not allowed any desserts. So the lambdas do not have desserts. And just to show you the menu again, this is the heavy chain menu. It has 40 things to choose from. He has 27 desserts because he's heavy, he has loads of desserts to choose from. And he has the adult menu because he's a heavy drinker, so he's allowed to choose beer as well. So he has the extra option of having beer on his menu, whereas the others, they're light drinkers. So in the juice menu, they only have 5 choices as opposed to 6. So that's how I remember the numbers: 40, 30, no desserts because you're light. And then because they're both light drinkers, you both have the same juice menu with the exception of alcohol. But the heavy guy, he's a big guy, so he's allowed a lot of starters, a lot of desserts to choose from, and a lot of juices plus the alcohol. Okay, so essentially the same thing is going to happen with your light chain. So you have your body decides for this particular cell, it's going to randomly choose a lambda chain. So of the 30, it's going to choose, for example, number 24, and it's going to obviously no D region, and it's also going to choose number 3 on the juice menu. Marry them together with recombinase enzymes, and those 2 together are going to code for the CDR3 in the light chain. And then the heavy chain will also be coded for by a different VTJ recombination in chromosome 14, and therefore we're going to get like loads of possibilities of things that can be variable. These fit together, and we've now got a really specific shape to an antigen that could by chance happen to fit into it. Okay, so we've now made the variable CDRs to make that variable region, which is essentially what we've done with the VTJ recombination. Remember, there's no D genes in the light chain, and the light chain can be either kappa or lambda. It's not that deep. It's essentially going to look the same as this, but essentially there can be different options as well. Essentially the menu is slightly different. Okay, the variable region, it can undergo some mutations. So for example, when the recombinase enzymes are adding stuff together, the recombinase, okay, so these variable regions here can also undergo some slight modifications. So either through adding or deleting bases, base codons, and that just occurs randomly and makes them more diverse, and that occurs in the bone marrow. We also have a thing called somatic cell hypermutation, which occurs in the lymph nodes, and that can kind of mutate these and introduce changes in that. But essentially, once those 2 have happened, it's going to become set. And once it's set, the concrete set is no longer going to be able to change its variable region. So say we've got this now, and we've done our VTJ recombination, and we've done our mutations. We've now got a specific shape for this antibody receptor, and this is going to live on our B cell forevermore. However, the thing that we haven't talked about is this heavy chain, well, not the heavy, the constant region, the FC region. And the FC region is able to change slightly, and that occurs due to splicing. We'll talk about that later on in a different video. But the FC region is able to change, but the variable region, the FAB region, once it's made essentially, once it's set in stone, it's set. The FC region can change later on, and these allow the antibodies to have different shapes for humoral immunity. Okay, there's some questions to end on that you can do in your own time.